I really want to welcome you all to this distinguished lecture by Judy Dubno, who is the Distinguished University Professor at the Medical University of South Carolina. She is a research audiologist and vice chair for research at their Department of Otolaryngology. Professor Dubno has received many honors and awards, including the gold medal from the Acoustical Society of America in 2020, and also for her impactful contribution and advances to our understanding of auditory neuroscience, speech perception, and clinical rehabilitation. And she was also the president of the Association for Research in Otolaryngology in 2000. Professor Dubno has been a major influence not just on my own career, but also on growing the research infrastructure to better understand how hearing happens across the United States, the causes of hearing loss, and bringing in newly independent researchers into the field via NIH review panels, serving on the advisory council for the National Institute of Deafness and Other Communication Disorders, and especially on the board of the Hearing Health Foundation for over 20 years. Please join me in welcoming Judy to present her distinguished lecture that's entitled Age-Related Hearing Loss, Translation from Animal Models to Human Hearing. Judy.
I'd also like to thank all of you for inviting me to present this lecture today, especially Professor Steiger from the Department of Biomedical Sciences. It's a great honor to be in Omaha, and I'm interested in sharing with you the work that we have been doing for several decades. And I'll start first by introducing many of my colleagues. That's a picture that I found from a few years ago that has many of our colleagues. But I think you all know that research is a team sport. I don't do any of this by myself. I have surrounded myself with very smart people who do work in all areas of hearing. And as I hope you'll be able to tell as I go through the presentation, that we're covering a wide number of areas of hearing science. And it only can be possible to do that with a team, and our team is really the best. So I'll give you here an outline of what I hope to present. First, I'll give just a very brief overview of the area of aging and age-related hearing loss. Then I'll go through some of the changes that go on in the aging auditory system, and especially specific auditory pathologies. And then I'll introduce our longitudinal cohort study of age-related hearing loss that's been going on for almost 35 years. And then some work we've done to better understand the underlying mechanisms of age-related hearing loss, both in animal models and in humans. And then I'll wrap up with a summary. Before I get started, I do want to say that if you have pressing questions, something that you need clarification, if you have that question, it could be that many others in the audience do, feel free to raise your hand and interrupt. We can also take questions at the end, but if you really want to interrupt with a clarification, feel free. And enjoy your lunch also, by the way. So we'll start with an overview of aging. You all probably know this from your clinical practices, that the population is aging. We're projected to have by 2060 almost 100 million people over the age of 65. And with that comes the increasing prevalence of hearing loss. And so you see there, by the aging of the population, we're going to also have many more millions of people with hearing loss. And those estimates that the hearing loss caseload by 2060 is estimated to reach 42 million. This is just in the United States. So just in case some of you are not familiar with hearing loss in older adults, mostly what you will notice is a reduced ability to hear low-level sounds. But that really is just a very small part of the the effects of hearing loss on the aging auditory system. And mostly it's poor suprathreshold auditory function, because I'm speaking to you at a level way above your ability to hear even the softest sounds. So communication is really affected more by suprathreshold hearing, and that is the ability to understand, perceive complex sounds, obviously the perception of syllables, words, and sentences, prosody, which is the stress and intonation and voice pitch in speech, the emotional content of speech, and sound quality is very important. And of course, for all of you, appreciation of music is also affected by age-related hearing loss. And these effects are especially noticed by people with hearing loss in certain conditions, when there are multiple people talking, when there's background noise, such as in a noisy restaurant, in fluctuating backgrounds with reverberation. In a room like this, there's a lot of reverberation in this room, with distractions that often happens when multiple people are talking. You've all had that experience where you're in a group and somebody comes in and there's a distraction. That's very difficult for older adults to follow the conversation. When there are no visual cues, and we all noticed that during COVID when we were wearing masks and social distancing. This affected greatly people with hearing loss, because those visual cues were no longer there, and because we had to be distant, the level of the sound was reduced. So the other foundation that we see in hearing loss in older adults is the large individual differences. So everything that I described to you differs greatly from person to person. The severity of their hearing loss, the rate of decline in their hearing loss. We see older adults that maintain good hearing well into their 70s and 80s, and others whose hearing starts to decline when they're in their 50s or 60s. There's a lot of individual differences in communication abilities, in quality of life, and in the effects of hearing loss on health comorbidities. And this means that there is a need, there's even large individual differences in individuals' need for hearing healthcare services and in hearing assisted devices such as hearing aids and cochlear implants. And generally that means that hearing needs for each individual are unique. So a lot of what I'm going to tell you is for the average older adult, but keep in mind that this varies tremendously from individual to individual. So what other effects occur with age-related hearing loss? Now here the evidence is not as strong, but there is some evidence that hearing loss may be associated with higher risk for certain comorbidities such as depression and anxiety, low self-esteem, increased social isolation, stress and mental fatigue. There's a lot of interest in a possible connection with cognitive decline and dementia, but the evidence is not quite all there yet. And there is some evidence for reduced mobility, frailty, and disability, and increased falls. So the effects of age-related hearing loss go beyond communication and sometimes will have an effect on health comorbidities. There is also some evidence related to lower income and employment and perceived social and emotional loneliness. That was also evident during COVID when we were separated from our friends and family, and we had fewer interactions. We noticed that an effect more on people with hearing loss. So in terms of these health comorbidities, and also the need for individuals with hearing loss to get the health care that they need, this is a public health concern. There's a significant unmet need for hearing health care. Many adults do not have access to hearing health care or cannot afford it. Most adults are not asked about hearing loss during their health care visits. They are not regularly screened for hearing loss, and they have not had their hearing tested. And most evident that only a small percentage of older adults who might benefit from hearing aids have access to them or use them. There might be a change on the horizon because there is now, the FDA has changed their regulations, and now there is an ability to get over-the-counter hearing aids. This has just started, and the rollout of that and how it's going to affect healthcare delivery is not quite yet established, but it is something that may change some of these limitations. So now I'll just move on to talk a little bit more about the aging auditory system and auditory pathologies. So as you can probably guess, there are multiple risk factors, not only aging itself, but how much noise an individual has been exposed to, the kinds of ototoxic drugs that they may have taken over their lifetime, and some of these comorbid conditions that I mentioned. So because of that, there are multiple sources of auditory pathology. There are also genetic and environmental factors that interact and accumulate over the lifespan, and that again leads to these large individual differences that I mentioned. So because of that complexity, back at MUSC, even before I was there, my colleagues had the good sense to develop a program of research that included both a basic science component with animal models of age-related hearing loss, and coupling that with clinical science research that included large numbers of adults across the lifespan. And that led to an NIH-supported clinical research center, which we've been lucky to have funded by NIH we're now in our 34th year. And a lot of what I'm going to tell you about is work that was funded under that clinical research center. So the overarching goals is to translate results from animal models of presbycusis to results from older humans, differentiate effects of these pathologies on auditory function in humans, and then relate these differences to hearing health outcomes, including the ability to use interventions and benefit from interventions such as hearing aids and cochlear implants. So the animal models of age-related hearing loss that have been used mostly are the gerbil right there, and mice more recently. And these animals are raised over the lifespan in highly controlled environments. They are not exposed to any noise. They don't take any ototoxic drugs. Everything about their diet and their nutrition is controlled. Even the humidity in the animal quarters is controlled. And so that means that in these quiet aged animals, all of the anatomic and functional changes that are seen in these aging animals relate only to the aging process. And this is a way that we can disentangle aging from the environmental effects, which is something that we cannot do in humans. And that allows us to identify promising targets for intervention, and also hopefully developing strategies to prevent or delay the onset of age-related declines. So we have focused mostly on three major sources of age-related pathologies. This is a cross-section of the inner ear, and I'm going to highlight the location of where these pathologies occur. This is a very simplified view of this. So one we call metabolic, and this is focused on the lateral wall of the cochlea, and we see a degeneration in this lateral wall. The next is sensory, which is focused on damage to the damage or dysfunctional hair cells in the inner ear. And I have an asterisk there to remind me to say that that is not necessarily an age-related pathology, but in humans, because humans are exposed to noise and ototoxic drugs throughout their lifetime, we do see damage like that in older adults, but that's not seen in quiet aged animals. And the last is neural, which is in this area right here of the auditory nerve. This is primary neural degeneration. So now I'm going to go a little bit deeper into these three areas. I also am going to preview the fact that we are very interested in the fact that each one of those has a unique hearing loss pattern or phenotype. And one of the main questions we're asking in our program is, can we see these same patterns in human audiograms? And an audiogram is just a measure of hearing loss detection. So keep that in mind as we go through this, because eventually we're going to get back to human hearing. So in metabolic presbycusis, again, that's in the lateral wall of the cochlea. This is responsible for production and maintenance of the endocochlear potential, or the EP. That's a positive voltage of about 100 millivolts present in the endolymph of the scala media. That's in this area right here. And you can think of it as a battery that provides voltage to the outer hair cells, sometimes called the cochlear amplifier. And in quiet age gerbils, there is a systematic degeneration of this cochlear lateral wall, together with reduced endocochlear potential, reduced EP. So together with that, we see that in metabolic presbycusis, the cochlear nonlinearities are reduced but maintained. That's tuning and suppression and otoacoustic emissions. It reduces the cochlear amplifier gain, which increases thresholds, that reduces the ability to detect soft sounds. And this occurs more in the high frequencies than the lower frequencies. And it also reduces the activity of auditory nerve fibers, especially nerve fibers with low spontaneous rates. So this was discovered quite a long time ago by my colleague Rick Schmidt, who designed a very clever experiment that I'll just walk through very quickly. And what he was doing was measuring the compound action potential. So this is an electrophysiological way of measuring hearing in a gerbil, an anesthetized gerbil. So what he did, and this is what these audiograms, we can think of this as a gerbil audiogram. And these are the lower frequencies, and these are the higher frequencies. And in this audiogram, what's unusual about an audiogram is the y-axis is flipped so that better hearing is toward the top, and poor hearing is toward the bottom. So what he did was he measured in lots of gerbils. He measured these compound action potentials to get a gerbil audiogram in these quiet age gerbils. And then he plotted this so you can see the change in the audiogram as the EP is reduced. And you can see there's a very specific pattern to this. The lower frequencies are reduced less than the higher frequencies. You see this gradually sloping loss in the lower frequencies combined with a more steeply sloping loss in the high frequencies. So, and we see that this was associated with EP loss, but it is not associated with outer hair cell loss. So then he did the very clever part of this experiment is he took younger gerbils and treated them with furosemide, which is a loop diuretic, and it has the effect in a younger gerbil of reducing the EP. So here is a gerbil with everything else normal about it, except their EP declines. They're still young. They have all of their hair cells. They have all their auditory nerves. Everything about them is normal, except their EP is dropping. And what you see is a very similar pattern, this sort of flat loss in the low frequencies coupled with a sloping loss in the highs that systematically declines with the EP. So it basically, the EP loss or metabolic presbycusis defined the gradually sloping audiogram of older gerbils. So then we ask the question, does metabolic presbycusis define the audiogram of older humans? And I just plotted some audiograms of our older humans from our database, and you can see the similarity in older adults to the gerbil audiogram. So that's metabolic presbycusis. So now we turn to sensory presbycusis, and remember this only occurs when you add environmental exposures like noise and drugs. There is a loss of sensory and non-sensory cells, and there are threshold shifts of 50 to 70 dB in the high frequencies, a loss of the cochlear amplifier, a loss of nonlinearities. And I'm showing you, these are two sets of audiograms, again, from human subjects, very similar to what's seen in gerbils. But again, this is only after adding environmental exposures. And then this is not seen in quiet age gerbils. And then lastly, neural presbycusis. This is focused on the inner hair cells and primary auditory neurons. The sensory cells are preserved, So this is primary neural degeneration, not due to hair cell loss. This is an example from a young gerbil and a quiet age gerbil of the change in the auditory nerve. There's a loss of synapses, a loss of spiral ganglion neurons, inactivity in the auditory nerve fibers, and reduced responses at higher levels, but detection can remain normal. There is evidence in humans of this change from archival temporal bones, and you can see this. This is from a population study. You can see this even starting from our teens. We are very systematically losing ganglion cell fibers throughout our lifetime, and this is true even without hair cell loss. This was from a later study that did a population study like the one in 1978, but they removed all of the cases that had no hair cell loss to make it clear that this decline was not related to hair cell loss. So this is something that is kind of the march of time. This is going to affect all of us, even if we're not exposed to noise or not taking ototoxic drugs. So it can occur without threshold elevation. It affects coding at high levels and in noise. It does affect suprathreshold auditory function, and it may explain some of the individual differences I discussed. And there is some evidence that older adults are at a greater disadvantage from this loss of auditory nerve fibers. But mostly now I'm going to focus on metabolic and sensory loss. So now is the first step in our translation from animal models to human hearing, because remember, the main question is, can we see these patterns that we've seen in quiet age gerbils? Can we see them in human audiograms? So as a start, I'm going to introduce you to our longitudinal cohort study, which is where we're going to get a lot of the answers about human hearing. And enrollment began in 1988. Right now, we enroll anyone over 18. When we first started, we were enrolling only people over 60. Everyone is eligible as long as they can provide measurable results. They're in good health so that they can come to the lab repeatedly, and that they have no evidence of conductive hearing loss or any active otologic disease. Then we bring them back to the lab every two to 3 years to repeat some of the protocol. And the protocol, basically we're measuring everything we can about human hearing in the auditory periphery and in the central auditory system, their ability to detect sounds, how their middle ear works, how their cochlear works. We do electrophysiological measures and behavioral measures. We test how well they understand speech, and in 1 or 2 ears, with and without noise. And we also ask them to complete lots of questions about their hearing, about their family history, about their own hearing history, their health-related quality of life, how much noise they've been exposed to, do they have tinnitus or ringing in the ear, what kind of prescription and over-the-counter medicines they take. It's quite an extensive protocol. And we measure, more recently, we've been measuring cognition, and we also do some structural and functional neuroimaging. And we also take blood. We are working, which I'll tell you a little bit more about later. We are looking at DNA extracted from blood to identify genes that may be under or overexpressed with age. We have also measured lots of clinical chemistries from their blood. We're not doing that anymore. We actually found several years ago that many were not related to hearing, so we stopped taking all that blood from our participants. They were pleased. And we also have a temporal bone donation program. So our participants can donate their temporal bone after death. And this has been a very important aspect of our program because of where the inner ear is located. It's not accessible in a living human. So really, the only way that we can make these structure, other than with animal models, the only way we can make this structure function association in humans is through their temporal bone. Okay, so currently we have enrolled almost 1,800 participants. We keep the number of active participants about 300 so we can get them in when they need to and they don't have to wait. 68% are over 60, but we have an age range from 80 to over 89. A little bit more than half are female. About 20% are from racial or ethnic minorities. We have almost 25,000 audiograms, lots of word recognition scores, and longitudinal data through 28 years. And we have over 1,000 DNA samples, and about almost 900 have undergone whole exome sequencing. This is what the audiogram of our at baseline, what the audiogram of our participants looks like for females and males. And again, better hearing is toward the top, poorer hearing is toward the bottom. These are the lower frequencies, and these are the higher frequencies. And these data indicate this is the approximately the lowest level sound that these individuals can hear. And you can see already that there's a difference between males and females. And if you remember those gerbil audiograms, we're already seeing what looked like kind of the flat loss in the lows coupled with a sloping loss in the highs. And again, these more steeply sloping loss in the high frequencies for males. The other lines are from two large-scale epidemiological studies that you may have heard of. One is the Framingham Heart Study, and in one of their waves, they also measured hearing many years ago. And the Beaver Dam cohort is from a study that is going on in Beaver Dam, Wisconsin, that is measuring everyone in the town. These are more traditional epidemiological studies of hearing, which ours is not. Oh, I'm sorry. But we were very pleased to see that the audiograms of the participants in our study matched very well with these large epidemiological studies. So this is the same data, but showing differences. Now, these are cross-sectional differences. These are not the same people in each group, but again, you can see a little difference in the pattern, and you can see this change in the low frequencies and the sloping loss in the high frequencies, and a little bit different pattern for males. And then I just wanted to, for those of you that maybe are not familiar with why I keep talking about this as a longitudinal study, in our study, the same individuals come back and we measure their hearing over many, many years. And the reason why this is so important and provides such strong evidence is that it minimizes the effects of differences in the individuals in their noise history, in their occupation, in their nutrition, any sort of generational changes, any pre-existing health conditions, that those cannot be teased out, or they're teased out with a lot of difficulty in cross-sectional studies. It provides evidence about exposures that precede hearing loss, and you can estimate the rate of change in hearing in an individual rather than just in a group, and that provides a lot of very important evidence. So there are some downsides to a longitudinal study. Data collection obviously takes many years. People have to remain in the study for many years. That makes recruitment a lot more difficult. There is a question about selective attrition in studies of aging. I mentioned that the people have to be healthy enough to come to the lab repeatedly. So we worry that the people that we're getting who are staying in the study for a long time are the healthiest individuals, and that means that we're actually looking at the highest performing older adults. And so we do tests in the database to make sure that is not occurring. We count the number of comorbidities, we keep track of their health, and so far we have not seen the selective attrition in our older subjects. And then of course the study is quite expensive. So maybe for these reasons, there are actually very few population-based longitudinal cohort studies in hearing, and really that's made it very difficult to isolate the effects of hearing loss from the effects of aging. So we're glad that we've been able to do this for 34, 35 years. Everything has to come to an end eventually. So we're trying to get the most out of the data that we have collected. And I'll just give you one very small example. So these are, this looks like an audiogram, but what this is actually plotting is the rate of change. This is the dB loss, the change in hearing per year. And so you see it's about 1 dB per year, or about 10 dB per decade. And you see there's a little bit difference in lower and higher frequencies, but overall it's about 1 dB per year. But there are differences between males and females. So around the time that we were working on these data, we noticed that there were a few papers coming out that suggested that people who were exposed to noise in their early life, that the change in their hearing over time was actually accelerated, that their hearing became worse at a faster rate than people who were not exposed to noise early in their lifetime. And there was a lot of interest in this because the conclusion before these studies came out was that the effects of noise on hearing, there is damage that occurs, that as long as you're taken out of the noise, or the noise stops, the damage stops. So the noise exposure stops, the damage that you have incurred stays, but it doesn't become worse once you're taken out of the noise. That was the understanding of how noise exposure and hearing worked. So their conclusion that a history of noise exposure accelerates the rate of change was kind of a surprise. And this was done in animals, I should say. This was an animal study. So we realized, well, we have the information in our database because we keep track of noise exposure. We know if they've been in the military. So we could look in our data and see if we get the same results. So at that time, we compared the rate of change in thresholds for people who reported a positive noise history and those reported a negative noise history. So as expected, individuals with positive noise history had poorer hearing, which is what you'd expect, especially in the higher frequencies and especially for males. But when we compared those with a positive noise history to those with a negative noise history and looked at their rate of change, there was no difference. So what we saw in our human studies is that there was not an accelerated rate of change for ears with a positive noise history. And that was true for males and females. So in our data from human studies, we did not see any evidence of continued injury to the ear after the noise exposure stops. We are still looking at this question because it is still, even though if you notice the year of that study, it was quite a long time ago, but it is still a very active question in our field. So we actually now have a lot more data than we did in 2005. So at this moment, we're actually repeating this analysis on all of the data that we have, and we have much more longitudinal data now than we did that. So, so now I'm going to transition to the question about, do we see these metabolic and sensory components in our human audiograms? So we're going to move a translation from the animal models to our longitudinal cohort study. And so you remember those shapes that I showed you for metabolic and sensory. And so we created these templates from the animal models and drew out on an audiogram what we thought a human audiogram would look like if their underlying mechanism was due to a metabolic component or a sensory component. And then, of course, because these are humans, we understood that there would be both. So we have another template of metabolic and sensory. So it has the characteristics of the metabolic component in the lower frequencies and the characteristics of the sensory component in the higher frequencies. So then we asked the question, are these phenotypes seen in audiograms of older adults? And do the phenotypes correspond to your expected demographics due to age and sex and noise exposure? So we searched all the audiograms that we had at that time in the longitudinal cohort study, and we used a machine learning tool. This was way back. So it was we could do this a lot better now that we have ChatGPT, but back in the olden days of machine learning, we assigned each of the audiograms to a phenotype, and we also had experts analyze the audiograms and categorize them. And the machine learning tool categorized the subjects with a very high degree of accuracy. We also then looked at the actual audiograms of the people who were assigned to these categories. And so those solid lines and symbols are the thresholds of the individuals who were assigned to these categories. So they fit the patterns very, very well. So an individual who is categorized as having a metabolic phenotype, their thresholds fit very well into this template. So we thought this was promising. But further, we also noted that the phenotype classifications matched the expected demographics. So for example, the people with older, or what we called at the time pre-metabolic, they tended to be younger, they tended to be female with a negative noise history. People who fit in the metabolic category were older, female, also with a negative noise history. The people in the sensory phenotype were younger, tended to be male, and had a positive noise history. And then finally, in the last category, they were older males with a positive noise history. So it all sort of made sense. So we then further validated this approach. We had longitudinal data, and we hypothesized that as people got older, they would more likely transition to a metabolic phenotype. And with my colleague Kenny Vaden, we were able to test that hypothesis. And when individuals changed over their lifetime from one phenotype to another, they tended to become a more metabolic or metabolic plus sensory phenotype. So that was one validation. And then we looked at other measures of auditory function. You may remember at the very beginning, I mentioned that certain noise exposures and certain changes in the auditory system affect otoacoustic emissions. And so because of the strong connection between that measure that we do clinically, we thought we could see if we can see the same characteristic in a measure of otoacoustic emission, and we were able to see that as well. So this is something that is separate from the audiogram. It's another measure of auditory function, and we see these same patterns in the same metabolic and sensory phenotypes in this non-auditory threshold measure. So that was encouraging. So we were able to validate this approach in several different ways. And then we decided we could do better. We had these 4 phenotype categories, but we kept facing the fact that most humans fell into more than one category because most humans have multiple pathologies. And we found that having these 4 buckets was just not sensitive enough to differentiate among individuals. It was just too gross of a measure to have these 4 categories. So again, with my colleague Kenny Vaden, we came up with a way of, instead of categorizing them into these phenotypes, is to actually calculate the amount of metabolic and sensory component in the audiogram. And we did this in all of our older adult audiograms. Now we have a continuous measure rather than a categorical measure, and we found that we were much more sensitive to individual differences. So I don't have time to explain how the calculation is done, but I'll just give you an example of what this looks like. So here is an older adult from our database. This is a female, age 73. The filled circles is this individual's audiogram, and the calculation that we get says that metabolic estimate is 28 dB, and the sensory estimate is 0. So that person is actually fully metabolic phenotype. This is a male, age 56, and we see just the opposite. We see a metabolic estimate is 0, sensory estimate is 28. So this person is a pure sensory phenotype. But most individuals were like the last one. Most individuals, this is a male, age 64, the metabolic estimate was 17, the sensory estimate is 28. So a little bit of both, and most people fall into this category. So then this is very recently we did this calculation on all of the audiograms in all of the baseline audiograms in our database. So let me just walk you through this. This is the sensory estimate on the y-axis and the metabolic estimate on the x-axis. So, and the gray values are anyone whose combined metabolic and sensory component was less than 20 dB. So this gray corner are basically people with generally normal hearing. And the rest, the shading, the red shading is the amount of the sensory estimate, and the blue shading is the amount of the metabolic estimate. So people who fall right on this line have various amounts of sensory hearing loss and no metabolic hearing loss. And people who fall on this line are all metabolic hearing loss and no sensory hearing loss. But you can tell that there are fewer of those on the axes. Most of people fall closer to the unity line. And so these are people with mostly sensory loss and a little metabolic. And these are people with mostly metabolic loss and a little sensory. But you can see how much more sensitive we can get with this measure rather than the 4 buckets that we started with. By the way, the dots are people who didn't really fall into one category or another, and there are a few of those. So, so what did we do next? So we looked to see whether we could see any relation of having a metabolic loss versus a sensory loss on other measures of auditory function. The first thing we looked at was whether they changed with age. We would predict that the metabolic component would decrease with age, and we could see that it does. The sensory component does not decrease with age because, as I mentioned several times, it's not really related to age, it's really related to environmental exposures. So then we looked at speech recognition. I'm not going to go through the detail of the measurement, but in this data, poorer is lower on the axis. So you can see the metabolic declines. The speech recognition is actually poorer with more metabolic loss and not so much with sensory loss. This was actually a surprise. I was actually thinking that the result would come out the other way. I thought it would be more damaging to have sensory loss, and therefore you would do worse with speech understanding. It actually was the other way around. So the data continue to present us with surprises. And then this is a very recent study that we're doing, an ongoing study right now, that we decided to look at a completely independent cohort. And we are collaborating with our colleagues at Boys Town National Research Hospital. And the study is actually related to another kind of otoacoustic emission. So the reason why the subject numbers are smaller, because in these data sets, the participants had to also have a measure of otoacoustic emission. And so that decreased the sample size a little. And so, so our Boys Town colleagues, that's Steve Neely and Sarah Harris, they collected all the studies in their databases where they had an audiogram and a measure of otoacoustic emissions. And we did the same in our database. And these are, so now we're comparing Omaha older adults and Charleston older adults. So we tried to make this as, as new a group as possible. And here are the 2 sets of audiograms. There are a little bit differences. The Charleston people have a little bit more higher frequency hearing loss. And also our group was slightly older than the group in Omaha. But you can see the pattern of metabolic and sensory components is very similar. So in their data, they had results only from one ear, from the better ear, and we had both ears. So in order to make it clear which is which ear, the symbols in this are the worse ear. So if you compare just the filled symbols in both, you're comparing better ear Omaha and better ear Charleston. And then the worse ear fill in a little bit more, but it's surprisingly similar. Now, I don't have there's a lot of other results related to the study, but I don't have time to give them to you right now. But we're I think serving as a replication cohort, we were encouraged that the results are very similar with an independent group of participants. So then we wanted to continue the replication, and we decided to go back to the gerbils. So in this case, we have translation from human hearing to the animal models. We wanted another level of validation, and we went back in our archives and looked at all of the data collected from all of our gerbils over the years. And we had quiet age gerbils, and we had the furosemide gerbils that I mentioned earlier, and we also had noise age gerbils. And we decided to measure the metabolic and sensory components of these gerbil audiograms. And so here is a gerbil audiogram from a quiet age gerbil, and you would expect that result would be all metabolic estimate, and it was 27 dB metabolic, no sensory. Here is a furosemide animal. This is a young animal exposed to furosemide. Again, we would expect that to be all metabolic, and it was. And then we have a noise age animal. Now, this audiogram looks a little strange because the noise exposure was right in this area. That's not exactly typical for humans to be exposed to such a narrowband noise. And in this case, we see both. We see some metabolic estimate because this is an older gerbil, and we see also a sensory estimate. So that we felt was another validation. And then we did, all of these gerbil animals also had an estimate of the endocochlear potential. So we had hypothesized that the endocochlear potential would change in the same way as the metabolic component. Again, this is flipped. So this is better hearing up here, poor hearing here. And this is the a high level of endocochlear potential is good. So you can see as the endocochlear potential declined, both in the quiet age gerbils and in the furosemide gerbils, the metabolic loss increased. So this was another validation that this approach was working. So then our last bit of validation, we're going to go back to humans again. And in this time, we're going to look at human temporal bone histopathology. And for this, we use the case reports from the National Human Temporal Bone Registry. We looked for cases that had an audiogram and a clear diagnosis of either stria vascularis damage, that would be right in here, or hair cell damage. And again, and in this case, we actually found one where there were longitudinal audiograms. So you could see this is a case with just metabolic damage to the lateral wall, to the stria vascularis, and no hair cell damage. And we see metabolic estimates changing with age and no sensory estimate. And then the other way, we have a case with a normal lateral wall, but hair cell damage, and we see the reverse. And so this is human data. So you could see that we kind of go back and forth in our translational approach. Sometimes we start with humans and go back to gerbils or mice. Sometimes we start with gerbils and go to humans, and then also human temporal bone. So, and our last, I mentioned that we are extracting DNA from our participants. We have done this in 532 of our participants. We have whole exome sequencing, and we have some very interesting preliminary results that I can't be very specific about because it's just preliminary, but we have identified some variants that appear to be linked more to sensory or metabolic loss. Some found only in males or females, and some are protective. We found some variants that are associated with better thresholds. So that's very promising. We're also comparing to data from large cohorts from the UK Biobank and also from an NIH database called All of Us. And this is in collaboration with our colleagues at King's College London, who are hearing geneticists. And so I'm going to summarize. What I hope you will take away from this is that audiograms from middle age to older adults appear consistent with predictions from animal models associated with metabolic and sensory pathologies. We've been able to classify and estimate metabolic and sensory components consistent with predictions based on thresholds, age, sex, and noise history. And we validated with animal models, other auditory measures, human temporal bones. And we think that, although there's a lot more work to do, that declines in auditory function may differ depending on magnitude of metabolic and sensory components. We see it so far in otoacoustic emissions and in speech recognition, but there are a lot of other questions we can ask about differences in this underlying pathology. We hope this will ultimately support a more personalized approach to auditory diagnosis, and hopefully may relate to success with interventions such as hearing aids. And lastly, I hope you will take away that longitudinal studies are important across the lifespan. We can identify modifiable risk factors. We can allocate to different risk factors and then separate the effects of aging alone. But it's very important to have large, diverse, well-characterized study populations. And lastly, the strongest evidence is when you combine human studies with basic science research. So I want to go back to my colleagues who are critical to everything I presented. I couldn't have done anything without them. So thank you for your attention, and I'm happy to take any questions.
Thank you, Judy. That was tremendous, and the latest data that you have from the last couple of years is really powerful. So thank you for that. May I ask if those who are on Zoom, if there are any questions from the Zoom, if you want to bring them through, Sean Lee, that's great. I do have one question while others pull their gather their thoughts together. You showed very interestingly that those who were not exposed to environmental factors like noise, that they were able to retain their hearing late into the lifespan. Why do you think human hearing is so much more resilient than in aging animal models?
Oh, well, let's see. You're talking about the. So I don't think I said they were able to retain their hearing. I think their rate of change of hearing. Now maybe it's similar. Was not affected by whether they were exposed to noise early. So you're asking why we didn't see the results that other people have seen in mice. Well, there's a lot of speculation as to why that is. One is that you might remember that the picture that I showed you with the very unusual audiogram of the noise-aged animal, where it had that very, I said, well, this animal was exposed to a noise, a narrow band of noise at 3 kilohertz. This is very unusual, and humans don't get exposed to that kind of noise. Well, that may be part of the problem, is that mice in the studies that are looking at this are often exposed to the kind of noise, and maybe at levels that humans are not typically exposed to. We're usually exposed to broad bands of noise. Maybe in the military there are unusual kinds of noise exposures, but typically a noise exposure in a very narrow range is not what humans are exposed to. So that may be one difference. There is some speculation also that the way the noise is calibrated, mice have very tiny heads. And so you have to kind of adjust the level that a human is exposed to with the level that a mouse is exposed to. And those levels may be very different. So the direct comparison of the studies with mice where aging effects are accelerating may not be quite the same as humans are exposed to. So that may be part of the difference. But it's still an open question.